Hello, everyone. First of all, I would like to thank the organization for giving me the opportunity to present our work here today. I'm really excited to present our work on graph-based pangenome analysis of a haplotype-resolved genome assembly of Petunia hybrida. My name is Peter van Dam. I'm a bioinformatician working at Genetwister Technologies. And the first slide is a bit of an introduction to Genetwister. We are a research-focused plant biotechnology company based in Wageningen, the Netherlands. And we work mostly for 5 plant breeding companies located in different parts of the world, shown here on the right on the map. We help them to breed for complex traits by applying the latest technologies in the field of genomics. We generally have 2 types of projects with them. First of all, bilateral projects with a clear applied research question in mind. For example, genotype-phenotype association analysis of complex traits or germplasm characterization. Other projects are part of our pre-competitive R&D program, where we identify new tools and technologies that can advance their breeding programs. The work that I'm presenting here is part of the R&D program. The species of interest in this project is Petunia hybrida. It's a very popular bedding plant, and the genome contains 7 chromosomes, and the haploid genome size is estimated to be 1.4 gigabase pairs. Well, as the name implies, it's a hybrid between 2 species, Petunia axillaris and Petunia species from the Integrifolia clade. Axillaris is located here in the tree, and Integrifolia is this clade here. The hybridization event or events occurred relatively recently, only about 200 years ago. And we will also see that from the results later on. In 2016, a paper by Bomberli et al. Was published in which genome assemblies of ancestors Axillaris and Inflata, which is one of the Integrifolia species, it's located here, have been described, although these are still quite fragmented. So to explain a little bit more on our motivation for this research, first of all, a high-quality reference genome is a very useful tool for Petunia breeding for mapping, marker calling, and other types of analysis like studying genes, regulatory elements, etc. And as I'm showing here in this box here on the right, where previously we would create a single linear reference sequence, which is actually a mixed representation of both parental haplotypes, indicated by the colors here, current tools and technologies allow us now to build a haplotype-aware assembly. This provides a more accurate and complete overview of the allelic variation in the genome, especially in highly heterozygous species, which is often the case in ornamentals. Also, it allows for following alleles through breeding programs and studying ancestry. Finally, we can then integrate different haplotypes and genomes into a pangenome and study variation and synteny within and between different genomes. Now, there are several methods for building haplotype-resolved assemblies. And in this project, we are using 2 of these methods and trying to compare the results. For both of these methods, we used PacBio HiFi long reads for sequencing the individual of interest. The first approach is trio binning, shown here on the left, which uses Illumina sequencing of the parents to assign the offspring reads to a parental haplotype, after which they are assembled separately. The other approach is using proximity ligation procedures such as Hi-C. After building haplotics using PacBio HiFi reads, the Hi-C data extends these into longer haplotype-resolved scaffolds. The experimental setup that we used in this project is shown here on the right. The plant material of the trio was provided by Sakata. And what you can see here is the 2 parental plants and the single individual that we sequenced with HiFi sequencing. So as you can see here, we sequence Illumina data for both of these parental plants, HiFi data for the F1 offspring, and then also Hi-C proximity ligation data for the F1 offspring. We performed an in silico experiment where we combined the Illumina data of both parents because we wanted to assess how different the haplotypes in the trio were. From the 4 peaks in this plot, you can see that the 4 haplotypes in the 2 parents are quite different, which is a great starting point for building haplotype-resolved assemblies. Previously, the best assembly results were, of course, obtained using an inbred variety, which is not always available. Assembly of highly heterozygous species typically resulted in highly fragmented assemblies. We can now actually make use of this high heterozygosity to separate the haplotypes and do very cool things with such assemblies. Using only the HiFi data, we performed a de novo assembly using HiFi-ASM with unzipped settings, shown here in the bottom of this slide. For comparison, the statistics of the Axillaris and Inflata assemblies are also shown, which are quite fragmented. Using HiFi-asm, we obtained a highly contiguous assembly already with an N50 of 100 MB and half of the assembly in only 8 contigs. We also tried the LGA and HiCanu assemblers, but the HiFi-asm resulted in more contiguous assembly with shorter runtimes. From the assembly size and the Busco scores, we can see that many of the haplotypes are separated in the assembly and that there's a high duplication rate, which is to be expected seeing the total size of the genome assembly. However, we don't know which of the contigs belong to the same haplotype and to which haplotype they actually belong. Therefore, we proceeded to try to separate these haplotypes into two. The first assembly just makes use of PacBio HiFi data with slightly different settings to HiFi-asm. The total size is very close to the expected genome size, and as you can see in this slide, typically haplotype A seemed to have a higher number of contigs than haplotype B, and this is largely to do with the mitochondrial and chloroplast contigs that are also largely assembled in haplotype A. Because the HiFi-asm plus the Hi-C data assembly showed the best results, we proceeded with this one and made it into a final assembly. The chloroplasts and the mitochondria were reconstructed, remaining contigs were placed in chromosome 0, and remaining plastid contigs were then excluded for further analysis. We now ended up with 7 full-length haplotype-resolved chromosomes, which show high completeness and a low number of duplication. And there's for each haplotype a small chromosome 0 of 23.8 and 11.6 MB respectively. We aligned the haplotype-resolved assembly to the Petunia axillaris genome, and as mentioned in the beginning, axillaris is one of the ancestors of Petunia hybrida. So for alignment, we actually scaffolded the axillaris genome using our hybrida assembly. And you can see that both haplotypes are present in the alignment, and that for many chromosomes, haplotype A is more similar to axillaris, indicated by these green diagonals here, suggesting a limited exchange of genomic content between axillaris and integrifolia since hybridization. Interestingly, if you compare the two haplotypes side by side, so Petunia hybrida haplotype A on the x-axis and haplotype B on the y-axis, you can see there's a separation and sort of compartmentalization within the genome. This is nicely illustrated if you look at the comparison between chromosome 4 and chromosome 2, which is shown here, where chromosome 2 has a much lower similarity score or identity score and a large rearrangement in the middle. And chromosome 4, as well as 5 and 6, seem to be much more similar to each other between the two haplotypes. We then proceeded to look at Synteny using a graph-based pangenome approach. So beside the value of high-quality haplotype-resolved assemblies, we can also use these to study synteny and structural variation within and between genomes. And we are applying a graph-based pangenome approach to do this, and we are using a tool called PanTools. This is a tool developed at Wageningen University by Eve Jonkeer and his colleagues. And we perform gene and repeat annotation. And by using PanTools, we build a pangenome including these 4 assemblies shown here on the left. So haplotype A and B from Petunia hybrida, as well as Petunia axillaris and Inflata, that were actually scaffolded using these haplotype-resolved assemblies. This graph shows the hybrida assembly with both haplotypes of all chromosomes, and the colors here indicate genes and whether they are core, occurring in all species, shown here in green, accessory in hybrida and one of the other species, shown here in blue, and unique, occurring only in hybrida. We can then also zoom into more detail in specific chromosomes and compare haplotypes based on synthetic genes. In this slide, the top shows haplotype A and the bottom half shows haplotype B. The line graph at the top and bottom shows the gene density in black and the repeat density in red. The bar below shows representation of genes in the pangenome, similar to the previous slide, and the blue and black bar below it shows the presence of genes in the opposing haplotype. Black genes occur only in the current haplotype, and blue genes occur in both. Then in the middle, there's an alignment block that shows the synteny between the two haplotypes, where blue shows synteny in the forward direction and red blocks in the reverse direction. What you can see here is a rearrangement similar as what can be witnessed here in the dot plot alignment on the left. Another interesting example that is worth highlighting is this black block here and here that shows a set of genes present only in a haplotype A. From the repeat content, we can furthermore see that the centromere is located around 85 MB, and the peak at 45 MB on haplotype B is interesting because the gene density here strongly increases and the repeat density decreases. There seems to be a block of haplotype B specific genes there. Taking such a pangenomic approach on haplotype-aware assemblies can be used to answer all kinds of biological questions about relatedness, conservation, and ancestry. In conclusion, we obtained chromosome-level haplotype-resolved assemblies of Petunia hybrida, which to our knowledge is the best haplotype-aware assembly of an ornamental species to date. This research shows that high-quality haplotype-aware assemblies are much closer now. Where high heterozygosity used to be a complicating factor in genome assembly, we can now actually use this to resolve the haplotypes. And as I've shown, we can then use a pangenomic approach to assess questions related to synteny, conservation, and ancestry after that. I would finally like to acknowledge a couple of people that were involved in this project. So my colleagues working at Genetwister, both in the lab and in the bioinformatics department, the people from Socata who are so kind to provide the plant material, the people at Radboud UMC for helping with the hi-fi sequencing, and Eve and Sandra from Wageningen University for doing the pangenome analysis. And with that, I'm really happy to take any questions. Thanks.